I want to welcome you here, since this meeting is largely bankrolled by the CompBiomed project, but because of the interest there is in containerization and container technologies, we deliberately wanted to make this meeting much broader. So, notice one thing that I made sure Marco inserted into my slides at the last minute. My affiliations include the University of Amsterdam, effective from the beginning of last month. You could think of that as a Brexit mitigation strategy, and I'm deadly serious about it. I stand in front of you totally embarrassed by the shambolic state of UK politics and its complete inability to determine what to do next. This is deadly serious when you're trying to run several very large EU projects, the status of which is as uncertain as the politics in the UK. So we have to do what we can to save projects like this, and we have activities going on to do that, and that's one reason why I have this affiliation here. It's an appointment in the Informatics Institute, which is just over the road here, and it's in the area of high performance computing. So do remember that the EU has about 9 centres of excellence in high performance computing, and there's even now a coordinating body which is called Focus COE, and I'm centrally involved in that, the interests of which that particular body is to identify cross-cutting capabilities that are relevant to all people who indulge in things like high performance computing. And historically, the world was divided into 2. We had high performance computing launching jobs in splendid isolation onto big metal, and the model was wait your turn, get to the end of the queue and wait till your job gets executed, and it's therefore jobs that can only be run on such infrastructures, namely because they're so expensive. The interconnects are the order of the day, and you want to be able to show you can run jobs that exploit the entire connectivity. Otherwise the argument comes, why are we doing this? As people in this room will know, supercomputers today are not really getting faster, they're getting fatter. It's a human condition as well. Fatter, not faster. You know what I mean. They increasingly have huge numbers of cores on them, sometimes with accelerators. It's the number of those that's making the machines hit high performance scales, but the performance by and large of the individual cores isn't increasing. It might be decreasing because of energy efficiency considerations. So there's very, very few codes that actually deploy at the scale of a large machine, even at the petascale. The European Union's drive to the exascale, sometimes I worry, hasn't factored that in fully, because there's a very small number of codes that will in fact run completely at that level. But that's okay from our point of view, because large domain decomposition problems don't accelerate the rate at which you can reach interesting, shall we say, physics. In fact, quite the opposite. The larger the core count, and depending on the communication patterns, the further away you get from reaching scales that are of interest to us. You have to be able to bridge time scales as well, and that opens the door to the importance of components, multi-scale modeling, and doing much more interesting and varied things on supercomputers. So there is all of this here, but in this talk I'm really focusing on one particular area, but what I've said is pretty relevant to it. I press this thing. This is just to get the acknowledgements out of the way before I run out of time and forget. Lots of people contributing to all this, and a bunch of companies, some of which are doing work directly in collaboration. I seem to have left one of the important ones out, which is Janssen, which is actually a partner in CompBiomed. And then you'll see some cloud providers and other resource providers here. So in fact, in our interest, we scan between the kind of much smaller end and the monstrous end. In this project, which I will refer to, which is a DOE INCITE proposal, we're now running applications on Summit, which is top machine in the top 500, but we can only do that by using container technologies to deploy applications onto it. So I'm just going to talk about one area. I have to run through it. I know there'll be a small number of people in the audience who are familiar with this, so I can't talk about it in depth either because of the time. I just want to give you the sense of the type of applications that can be deployed either on very large scale machines and the benefits you get from those, or by allowing them to run in cloud environments, or of course combinations of them. And although our project is centrally about mechanistic and predictive modeling in medicine, as opposed to the giant wave of we'll solve everything by the triumvirate of BD, big data, /ML, machine learning /AI, artificial intelligence, we don't deny the relevance of that at all. So some of this stuff involves trying to commingle those things together. And again, the combination is relevant here. So basically, I'm talking here about something which is called BAC, Binding Affinity Calculator. The aspiration of this is easy to describe. It's a coin, in one sense, which has two sides or faces. One is to aid drug discovery, because the drug industry has a model that's totally broken. 12 years, $3 billion, one drug comes out of it. Wait a minute, we live in a post-genomic era. Don't we know that everyone could be different? Sure they are. How do you make more drugs? How do you make them faster? This is part of an attempt to address that. On the other side of the coin is clinical decision making. Imagine you have genomic information on your patients. You have a bunch of drugs, if you're lucky, that do exist, and the question is, which drug do I give to which patient? Turns out that the answers can be achieved at least at some level by using the same type of infrastructure. So this is telling grandmother to suck eggs if you're into the field, but otherwise not. Imagine a cartoon of a protein and a drug. If they're separate, they have some form of free energy. If they're bound, they have another. And this thing, called the binding affinity loosely, is what measures at first level in the search for drugs something important about the characteristics. So if we can get a handle on that, we've got a chance of doing things fast. And what we're after here is rapid, precise, accurate and reliable calculations, because our aim is to, as it were, take over the pharma industry, to stop them spending so much time on experimentation. At this moment in time, that's what they do, make a lot of attempted drugs and test them experimentally, and usually the theory on computation is just an afterthought. It needs to be in front to determine which kind of compounds they search for. In the clinical context, that's also very pertinent. So there's two types of calculation which I'm going to swing past you. One is, for the aficionados, I want to calculate the free energies of the components separately in a binding event like this, and I do it by calculating the free energies of each initial and final. That's called an endpoint method. And there's a bunch of ways to do this, and some of them are cheaper than others, and one of them, which we've developed, revolves around a technique which uses all-atom MD, molecular dynamics, which is computationally pretty intensive, and it then does some parts of the calculation using an approximation that the solvent around the protein is a continuum. The other one is, did you know that alchemy is back? It died out, 1,000 or more years ago with the Arabs, but it was about turning base metal into gold. On a computer, of course, you can do that. You turn one drug into another. It's not a physical event, but because the free energy is a function of state, it doesn't matter. We can go round a loop where we alchemically transform green to red for the drug, leaving the protein separate, and then look at the combination in the complex. And the relative free energy of the binding is then actually from the free energy changes associated with these two events here. That turns out to be computationally quite efficient, except it historically has been expensive. And here's the other side of this. Did you know that if you run an MD simulation, the next person who runs it isn't going to get the same answer? Basically, virtually no chance of that. This is just an illustration of it for one set of calculations. Can you imagine? These are all the 9 FDA-approved HIV-1 protease drugs. This is one component of the free energy. That's another, and it's histogramming the results when you run these calculations with different initial conditions. They're all over the place. And the free energy changes can be as different as 10 to 12 kilocalories per mole, which is just ludicrous in terms of accuracy, unless you take this into account and do the computation and averaging correctly. But what it means is, this is just like weather forecasting. I want to know tomorrow's weather today. That implies I run a lot of calculations. If you're aware of what happens in the weather forecasting industry, we have high-performance computers. You run a lot of calculations. You compute averages, and then you talk about tomorrow's weather in a probabilistic sense. We don't say it's definitely going to rain or not, but the outcome is actionable because I then reach for my umbrella ready for that event to take place. This is what we're trying to do in the medical context, whether it's this molecular end or cardio or anything else. We want actionable decisions. That means ones that are done before a decision needs to be made. That gives the emphasis to the speed at which we need the calculations to be done. And historically, you'd have a problem like this, where you have a bunch of so-called drugs or candidates, you run an MD simulation, and you try to claim you know how the predicted binding affinity varies with experiment. And very seldom have we looked at the errors and the reproducibility issue. Reproducibility now being a hot topic in all areas of science. So we must do better than that, and we do that with ensembles. The methods are twofold, this so-called ESMAX one. The other is a thermodynamic integration technique, the one I mentioned with Alchemy in it. And then we have a whole recipe of how we do these things. A lot of simulations for the replicas, specific protocol for how long the runs need to be done. And how many calculations you want to do also plays into the fact that these machines are now fatter, not faster, because we're in fact running lots of them. This is just a workflow of the sort you get, where you count up the hours depending on the state of your machine, etc., how quickly you can get an answer running lots of these calculations. Typically, you need tens of thousands of cores to get anything interesting happening. This is the TIES methodology, where again, it's just susceptible to quasi-embarrassing parallelism, because each of the individual calculations in the alchemical transformation is simply run independently on a bunch of cores, maybe a few hundred. So in the case of the large-scale high-performance computing, we famously took 2 days of the LRZ machine. It's actually 2 phases 1 and 2 of SuperMUC. They gave us the whole lot, which is 250 cores, 1,000 cores, for about 36 hours, and we ran a whole bunch of these calculations across the lot. So you get all kinds of benefits, combinations of drugs, proteins, investigated protocols, insights into methodology, and of course academically a lot of publications out of it. What do you need, though, for a properly integrated infrastructure, which is coming to the core of this? You need some kind of coordinating workflow engine, steps which involve the building of the models in the first instance. You've got a range of these drug candidates or molecules that have to be built to fit into your individual protein simulations. The simulations have to be fired off on some HPC or cloud resource, and afterwards, typically, you do analysis on some other location. So it's the linking up of all of those that are required, and has led us to invest time in a user-friendly form of the the binding affinity calculator that manages execution on a range of sources from HPC to commercial clouds, which is where the issue of their role with containers comes in. Just to whet your appetite, this is a sample of the kinds of machines we use at the high end for these computations. But if we're working with a pharma company, these companies have no access to those resources or under very controlled ones in a project like Comp Biomed. How are they going to do these calculations? Well, just briefly, on HPC, you have a deployment which goes through a client onto a server, and then something like the Radical toolkit that has been developed by a colleague of ours called Shanta NewJar, that can then deploy each of these workflows separately on the HPC resource. But if you're doing a commercial cloud, the benefit of this is, since these applications are not trying to use the whole of the infrastructure with the tight interconnects, you can potentially deploy this onto the computing resources they have. If it's Amazon Web Service, frankly, they're not interested in parallelism, but you pick it up for free if you have a 128-way node on the machine, or of course they deploy with great rapidity GPUs, which are very powerful for molecular dynamics. So this is an example of the cloud deployment that BAC uses, where under AWS control, it manages these instances to build and then run the application. Microsoft Azure is quite interesting to us because it's an HPC cloud. with InfiniBand interconnects between these nodes, which means they're more aware of the fact that you might want to run across multiple nodes. And there are some interesting benchmarks we have with NAMD. This is a molecular dynamics code running on the Azure cloud, where it scales pretty impressively if you need it. So coming quickly to the DOE project, the INSPIRE project, this is one that's in the context of patient-specific biomedical challenges. It's addressing a problem that occurs quite commonly in cancer, particularly the question of if a patient's suffering from a form of cancer where they've developed resistance to the drug you've been giving them, how do you then treat them, the second-order effect? And 90% of deaths from metastatic cancers arise from that context. So wouldn't it be a good thing if we knew how to treat it? And the major factor in some of these resistance phenomena goes through missense mutations, which essentially just means one SNP being flipped and leading to a single change. So the mutations are rare, and actually that does make it very difficult for the great machine learners to come in and say, don't worry about how this works, I've got all the data, I'll turn a handle. They actually can't do it, but their aspiration is to combine our methods with machine learning to predict the choices. And in this context, we've had a gazillion hours on the Titan machine that once was #1, and now we've migrated onto Summit, which is, with some interesting challenges. But for your information, if you're not aware of Summit, it's supposed to be there to work with machine learning and conventional HPC. Look at its description. It has a huge number of NVIDIA GPU accelerators, essentially on all the nodes, and its benchmark performance is that, which is pretty fast, but it gives problems to us. The workflow that we've developed here makes use of something called the OpenShift container as an applications platform here, because what we're actually aspiring to is to target hundreds of thousands to millions of molecules where we would do these calculations and machine learning. It's a loop which has machine learning on the patient data and the molecules, goes round a loop. When it needs to do calculations of the free energy, it fires those off, and they go back round this iterative process again. It involves quite a lot of steps and exploits the GPUs as well, but the problem with the architecture of Summit is it's sort of designed to just do monolithic applications, and ours is not in that category. So we have to look at what codes will run on it and where they will run, and we've had to kind of join up one of the machines called Marble that originally had nothing to do with Summit, which can deploy some of the components of the tooling we need in this pipeline. And then with the combination, some of them do not run on Summit, we can join them together and make this workflow come together. This isn't happened, well, I would just say it's on the verge of becoming possible now. And because I'm coming to the end of the talk, I'm just trying to emphasize the importance there of using the container technologies that we've had to deploy already in versions of BAC, which are used for the commercial activities, back into this high-performance computing environment to make the whole pipeline come together, to support the advances in the high-performance computing, and indeed the reproducibility of the calculations once they're done. With that, I should stop and thank you for your attention.